I think with sedphat. This is data that comes from the new machine. I found one of the original scans that um, Nilafar and I did when we were first getting to use this thing, and the data is the data is lovely. I mean, look at the resolution and the smoothness in the lines. The optics are just astounding in this brand new machine. You load it in the usual way. You load new data files. Let me see if I could find it. But this is one of the small things I wanted to show you. When you've got a bunch of pieces of data, you go to the file name and you can type in something really simple like for this data set, we got data on radial absorbance, which is these RA2 suffixes, and radial interference. And you want to load in either absorbance or interference. So the way you select one or the other, or if you have multiple cells that you're doing data collection on, you might want to put the number for the cell you actually care about in this last position. But asterisk.
Dr. Art, it's not updating your screen whenever it's.
Oh, let's try it this way then. Do you see the select files to fit now?
Yeah, I see it now.
So in this file name, you can type in things like wildcards from Windows. So like asterisk.ra2, if I put in everything, it'll show me the radial absorbance and the radial interference, but I just want to look at the radial absorbance data and I don't want to have to like shift click everyone. So I'll just click on the first one. I'll scroll to the last one, shift click to select all of them, click OK, press enter to see them. I won't load the previous analysis this time or worry about that stuff, only because I want you to see how nice this data looks if we zoom in a little bit on it. I think that's lovely personally. Um, the meniscus position, I'm not sure I really trust. I might scooch it a little bit more here. This first, you can double click and press enter to reset some of these lines. So now I'll be fitting the data between the green lines. The meniscus is in red, the blue line is at the bottom, but I'm not actually looking at that. Once I've loaded the data in, I go to C of S distribution, I click on parameters. I already know when we've looked at this before that 15 is too high for an S max. I think the viscosity should be more like 0.010. I set the confidence level to 0.68, the resolution to whatever. I'll let it fit a baseline. I'll run through the calculations. I don't know why it's so slow. I just did this and it was faster. And it seems to be okay. I couldn't get it to fit to a single species, which I thought would be an interesting thing to show, but I do want to spend some time on sedphat instead. So you get to see a species if you run it. Really, we should be fitting the data, but that's probably going to take too long. But there's a little streakiness in this bitmap. There's some trends in the residuals, which make me feel like I'm not really fitting the data right. So I wouldn't really put any weight on believing this particular molecular weight for this thing yet. But I wanted to show you what sort of data we can expect out of the new machine. The other important thing to say is that when you're doing an experiment and you're loading data in, this rainbow color, besides being just cool looking, it's important that you have a rainbow that accurately reflects the entire range. When you're running sedimentation velocity, you want to make sure that you spin things as long as you dare, meaning that if you don't need the protein back again, your best bet is to make sure it's a smear in the bottom of the tube at the end of the day. So you could do a run for much longer. The new machine is much faster at doing scans, so we asked it to do 100 scans in like 10 hours, and what it did is it did 100 scans and then it did nothing for the next like 6 or 7 hours when it was already done with 100 scans. If I had asked it properly to do like 400 scans over the course of 10 hours, I would have some data in this region here where you'd be able to see some of the effect of what the protein was doing at later times. And that gives you some more accuracy at actually calculating a molecular weight because you have more accuracy at getting a diffusion constant. So let's skip that for now. So any other questions on sedphat? Sedphat, you launch very much in the same way. If you have it normally starting, one of the things you can do, I mean, sedphat is a very similar piece of software. Um, there are a bunch of additional menu items at the top. Most of the time, you will find yourself loading in multi-speed equilibrium data, which means that you're loading in different sets of equilibrium data that have different velocities, but they're done on the same protein, the same concentration, the same cell, just sort of different days with different speeds. And the idea is that the radial position, the bottom, and the meniscus should all be about the same. The concentration, the integral under the curve for each of these things should all be about the same. So you want to load them into a single data set as opposed to fitting them as individual curves. If you've done that, and let's see if I can find a data set of multi-speed data that actually looks good. I have tons and tons of data. Um, you can do the same trick where, let's see which one this. I look at all of my data from this A series, and I relabeled some of the final scans. So um, these scans are presumably happening as a function of time every 4 hours. And I previously looked at the difference between successive scans to convince myself that yes, indeed, I'd come to equilibrium. And then I came along and I relabeled this last scan at the name of the speed. I think this might have been the one that I loaded in. If you say yes, you'll probably want to spend some time staring at this thing so that you set the experimental parameters up, but you can set it up again later if you like. I have the V-bar for my particular protein, the buffer density, the temperature. I'd read some of this stuff from the data file. I know it's a 6 center centerpiece, which has a, strangely enough, the path lengths are 1.2 instead of 1 centimeter. The units here are something you need to pay attention to a little bit. These are extinction coefficients in per molar per centimeter. These are meniscus positions in centimeters, which are almost always the same. Sometimes sedphat is nice enough to tell you what units things are in. The path length, though, is in centimeters. You really only have the one path length, so you don't have to worry about it too much. So this is now a multi-speed data set with multiple scans. I don't want to, what am I doing here? I don't want to do any of that stuff. And it's asking me to do a lot of stuff in terms of setting up concentrations for how much I loaded into the cell. And then it's asking you to save this XP file. And an XP file stands for experimental parameter file. It contains everything you need to know about the actual experiment. And I think what it's trying to do is get me to do an experiment. It's trying to load in an additional experiment. So let's just, I don't remember, I don't want to overwrite old stuff. So I'm just going to call this one temp.exp, although I suspect it's exactly the same as the one I had. You can go to display, you can change the data mark size so that things look a little bit different. It asks you which data set. So now I'm looking at two different data sets. It's basically the same data set, I just loaded in twice. If I wanted to, oh, that finally converged. Yippee. So if I wanted to, I could go to data set 2 and change some of the parameters on it and make the data points a little bit larger. I could set positions for things. If you use control click and enter, it will set things in a reasonable spot. It knows the sort of left side should be the meniscus and the right side should be the bottom. If you double click in the middles, it'll let you set the green things, the green parameters. Usually, once you've done loading in a number of data sets, there's a lot of other menu items you can deal with here. One of them is experimental parameters, and I can go to experiment number 2, and I can turn it off in terms of being active. It will remove that from my field of view, meaning that we're not going to use that in my current fits. So if you have multiple pieces of data and you're trying to fit them all at the same time to a single model, this is the way you would do it. But maybe you're getting weird results from one of your data sets and you have to think about excluding it from your fitting. So you go and you pick a model. Let's pick a very simple model, just species analysis, which lets me have a very simple global parameters. I can fit a single molecular weight. It's already given me a starting molecular weight. And usually you have to fill in something for concentration. Experimental parameters already fit. I already added information in. The run looks absolutely terrible, and I don't know why. What is it doing? It's probably in this that I've asked it to fit all of this stuff, and I don't want it to fit all these extra noises. So now I have something where some lines have been drawn in red and blue and yellow to try to calculate what the molecular weight is. And really, when you click run in sedphat, it's taken the initial value I gave it of 50,000 or 50 kilodaltons, and it just ran with that and said, okay, let's see what it looks like. So this is what a 50 kilodalton curve should look like, as long as you adjust the sort of y-axis so it fits. But that's not what I really want. I want it to tell me what the molecular weight it thinks things is. And so I've asked it now to adjust the molecular weight, and it's giving me crazy values. It says it's a kilodalton. So that's garbage, of course. And one way of saying that is that maybe it's just not fitting right. I really think that what I should be doing, instead of adding another species, is adding a couple other zeros here and trying to get this to fit to something that's more like 100,000 daltons. That's actually a little too high of a guess. I don't know why it didn't fit well with 60,000. I think there's something wrong with the way I've set up this particular experiment. I think I need to have TI noise on because I have interference data. I don't know. All right, that looks a little better. And so then if I fit this from 60,000 daltons, hopefully it'll give me lines. Really? That's totally bizarre. All right, well, it's misbehaving somehow. And so what I would suggest to you is that you end up spending a lot of time trying to get the software to do what you expect. I'm going to add in a second species and see if that makes things work a little better. It's going to take longer to fit. I don't know why it's trying to do things in the second data set, but that's going to be a problem. So basically what you do is I've loaded in 3 sedimentation equilibrium curves for a single experiment. It's probably going to take forever to converge. It might even crash. And I'll see if I can get it to stop. And sometimes if it's being really annoying, it's easier to just start another instance of sedphat. At least it is for me. Hopefully you can see the second instance. So what I did was I loaded in new multi-speed data. But once you've saved your data as experimental files, instead of loading in the individual data scans, if you've already set up your experiments and saved them correctly, a lot of times it's easier to save your data as XP files, which it prompted me to do when I saved the raw data. The XP files contain a little bit of extra information. It contains the last fit, it contains the meniscus and bottom positions for you, and it usually lumps together multiple scans at the same time. So here I have this particular scan that I was trying to fit before at 4 different speeds and as an experimental data set. It should look more or less the same as before. If I bump up the data size to like 2, it's the same set of data. And it should give me a better fit this time once I allow it to fluctuate amongst the different values for bottom and top. It's not. The nice thing is that if you want, I can load in a second set of data, which is, where's column B? So 3 speeds of column B. This is a different experiment now with a different loaded concentration. I can load up a number of these different data sets. I did this experiment 3 times with 3 different speeds in 3 different concentrations, and I called them A, B, and C. Now they're labeled as like experiment 1, which is A, experiment 2, and experiment 3. All 3 of them have different concentrations of material in it. But in each window, it's the same total loading. I have a problem in that I've been doing interference data for this one, so it ends up making fitting things a little bit tricky. If I go to my experimental parameters, I believe I need to leave RI noise on, the right type of centerpiece, and it will prompt me to go through all of my data sets, correct the problems that are there. As you can see, sedphat is a little bit more complicated than sedphat, and really all we've done in terms of modeling is that we've been exploring the species analysis. There's a lot of stuff you could do here. I spend most of my time doing species analysis with mass conservation constraints, which gives you a little bit more surety of the correct answer. And then instead of doing the C of S distribution, if you happen to load in sedimentation velocity data into sedphat, you can do this hybrid local continuous distribution and global discrete species. It's a mouthful. It's a model, but it's basically the C of S, but it allows you to fit a couple of discrete species as well. So suppose you have a contaminant that's like 100,000 Daltons and you don't care about it. You can fit that as a single discrete species, but then do a C of S on things that are smaller than 100,000 Daltons. And it's really convenient to be able to do more complicated modeling like that. Like it implies you need to know something about your system before you actually do that. And it's usually worth your time to do a sedphat analysis before you bring this into sedphat. But you can do much more complicated data fitting with sedphat. You can even get into things where there's non-ideal species, multi-wavelength considerations, two configurations, which I've never actually had to deal with sedphat. But a lot of times I'll find myself doing monomer dimer self-association. So suppose you want to figure out a dimerization constant. If you pick that model, the global parameters window is now vastly different. I usually turn on local concentrations for that, and I usually turn off fitting the molecular weight, and I'll turn on fitting the log Ka, the association constant in per molar. So this is the log Ka, so this would be, if I made this 6, then this would be saying that the per molar Ka is 10 to the 6, meaning that the Kd for dimerization would be 10 to the minus 6 or micromolar. And so that sort of thing you would probably be able to see in analytical ultracentrifugation, and you'd be able to fit it in this sort of modeling. In that case, though, I would not want to fluctuate the molecular weight. I would presumably know the molecular weight of my monomer with some sort of surety. And you should note too that in terms of this model of A plus A goes to a quote unquote dimer of A, if you have a protein that is a dimer and it forms a tetramer, and you know that you can see the dimer and the tetramer at the same concentration, then you should set your experiment up at that concentration and then still use this same relatively simple model, put in the molecular weight of the dimer, and then look at the Ka of the dimers forming a tetramer. So you could use the same model to look at higher order oligomers. A lot of these other things are generally not worth your time trying to mess with. It deals with a lot of non-idealities, and if you set the experiment up correctly, you have a better chance of actually not having to worry about that. I would encourage most people to try to turn on mass conservation as something that you can use. Mass conservation puts on a very strict constraint on how well the data fits. And it means that you shouldn't be creating signal from or losing signal from one place to the other. Now, if you're going to do oligomerization constants with your data, if you're trying to model this as a way of figuring out an oligomerization constant, one of the most important parameters back here in experimental parameters is the extinction coefficient. And you can see that you can actually vary the extinction coefficient by fitting as well. And I would encourage you not to do that unless you need to. There's a special reason to be able to do that. Most of the time, though, you should not be adjusting your extinction coefficient to fit your data. It should be a constant that you know. The only reason we actually do that is that, for instance, if you're trying to measure the extinction coefficient of a known concentration of a species, and you're measuring it at a wavelength where you don't know the extinction coefficient. So it might be worth trying to fit there. And there's examples for that on the analytical ultracentrifugation website. Not this workshop's website, but the one at analyticalultracentrifugation.com. So usually you would want to make sure you put in the correct extinction coefficient for whatever signal you're using, and whatever wavelength you're looking at, and whatever particular data set. But then when you go through and you've edited the extinction coefficients for all of your different species and all of your different models and all of your different experiments, then you can have a reasonable time and a reasonable chance of getting a good extinction, a good oligomerization constant for this monomer dimer. You can even see there's a lot more complicated models that you can play with, depending on whether or not you actually have that kind of stuff going on. You can imagine if you're trying to go monomer trimer, it's very similar to the monomer dimers global model. And then this happens when you're trying to do things and you have to force quit. Come on, darling. So that's loading in multiple data sets at the same time. sedphat actually lets you do a lot of different interesting things. One of the ones I wanted to show you is that you can load in your data this way. There's actually a lot of different kinds of data you could load in. You could load in sedimentation velocity data and fit it this way. You can load in DLS data, ITC data, blah, blah, blah. Some of the spectroscopy data is for things like fluorescence, anisotropy, or just straight up fluorescence, like from a FRET experiment. If you have this written out as a data file, and that describes the interaction between two parameters, then and you have other data sets, you can load in multiple different data types. That was the dream of sedphat eventually. And loading in complicated configurations of sedphat is done down here. So you could load in raw data in this section. You can load in individual experiments, which may be a compilation of multiple scans. Or you could load in a complete configuration of an entire preset series of things in sedphat, and I have one or two of those. That's actually a great way of storing your fits when you're trying to deal with things. That's cool. What happened here? Like my mouse has gotten slower with time. So here we have 3 pieces of data, a fairly complicated model that pops up from the configuration. But if you were to look at it, we have 1, 2, 3, 4, 5 experiments that were done with different concentrations, 3 different speeds, and that's why you have red, yellow, and blue. One is for each different speed. Red is presumably, or blue looks like it's the slowest speed, yellow looks faster, and red looks faster still. But the experiment was set up with 3 different ratios of A and B and 3 different total concentrations of A and B. Those things would need to be entered in parameters, and those things would need to be entered in the concentrations for all of those different independent species. If you pick the correct model, and you should note that this model has been pre-populated because the configuration that sedphat saved things as stores all of the information from your best fit model with all of the data in it, and all of the fitted parameters are all lumped into that one configuration. So when sedphat crashes, hopefully you've updated your configuration. You can actually go to data, and once you've got a configuration that you've saved it as something, every time you have a fit that makes sense to you, it's worth your time to either save it as a new configuration or to update the current configuration. So when you're trying on different models for your data set, you can actually do it that way and you don't really go backwards. Obviously, this is the sort of thing where you need to start writing down file names in your lab notebook so you know what you're doing. So we loaded it in a configuration for a fairly complicated model. Presumably someone was trying to figure out some interesting information. For this particular model, you'll notice that you can't even adjust the molecular weights. You're expected to put in a molecular weight that you know, total concentrations for these two, although apparently they're fitting them. That's a little bit of a scary thing to do. The main thing that they're trying to fit is formation of this complex and the equilibria constants involved in that. So if you were to just run it, the configuration was saved with the best fit parameters from the last fit. So of course the lines go pretty well through the data. You can actually click run or fit on this one and it will actually fit. It'll take a little while to fit. If you look up here, it has put in a bunch of the KAs, the association constants, for this complicated model of whatever it is. Um, A plus B goes into a heteroassociation, and then a second B binds. So it's a kind of a complicated model that's been done here, but presumably people have reasons to believe that this is actually going to be good data. Um, it it's a difference between modeling data where you need to know that it's worthwhile using this model. When you look at this data, I could easily fit a single or two species curve to a two species model to any one of these curves. So you have to be justified and somehow you have to have some other information that leads you to believe that there truly is this heteroassociation actually happening before you get into this kind of modeling. So does that make sense to everyone? And I just wanted to show you a little bit about what sedphat is really there for and what it's designed to do. You have multiple experiments. Each one of these are a sedimentation equilibrium run that was done with a different loading. And what they probably did is they varied the ratio of A to B with a total concentration so that they could actually see the experiment, if that makes sense. And as you'd expect, if you add more B, it would promote more formation of one thing or the other. If you explore the parameter space with enough different concentrations of A and B, you'll be able to back out the equilibria constants for this heteroassociation. So I kind of like this example because it's fairly incredibly complex, but otherwise the data itself, it just looks like a bunch of exponential curves, right? Like you could fit them to all sorts of really simple equations and come up with all sorts of different numbers. But the power of doing this globally with one model and 5 different experiments, each experiment has 3 different curves to it. Now you actually have some statistics behind you suggesting that you really do have a unique uh, association constant. Questions on that? So we'll see if sedphat crashes one more time for me. I was going to load in a different configuration as well. This is kind of what we would do if we were trying to fit sedimentation equilibrium data. Even if it was to a single species, you would just pick a simpler model, but you would still load in 3 or 4 or 5 different experiments, and your screen would look like this. When you publish this as a paper, you would probably only show 1 or 2 of the most representative curves. It just gets to be overwhelming otherwise. But in terms of being able to fit things rigorously, you want to say you went through all of this. So sedphat doesn't always deal with just sedimentation equilibrium is the fun thing. It actually deals with all sorts of different data types, like I said before. If I go to, where did these people do this? If I go to this data set, I think, and I pick up all the data, this is a sedphat configuration of all the data. And in this particular model, there's only 2 species, but what's cool about this configuration you'll see is that we have 3 different data types. In experiment 1, we have something that looks like sedimentation velocity, and it's got that jitter problem. So like these blue curves here, they need to be subtracted. They need to lose like 1 fringe value. You'll notice we're plotting fringes on this first experiment's y-axis. So these red curves should probably come up. These curves in the middle should probably come up. And these curves in blue should probably come down. When I fit it, I can subtract all of the systematic noise, which will account for the jitter. But the jitter is just something because you're trying to deal with the image of the interference data. You can imagine, though, that there's still an inflection point in each of these curves. It marches down radially as a function of time as you go from dark blue to red. And because we're trying to fit in 3 different data sets into one computer screen, the sedimentation velocity experiment is now squished. In the middle panel, we have sedimentation equilibrium, just a single curve. And in the right-hand side, we have dynamic light scattering. All 3 of these are describing a protein based on size, or maybe a protein mixture based on size. When you look at the species analysis and the global parameters, they put in some starting values. There's some little guys, and there's some not so little guys, 4,000 and 18,000 Daltons. That was the initial and best fit parameter for this. If you were to run this as a global run, and you could run one of these at a time. If you wanted to run just experiment number 3, you got a line through the DLS data. It suggests that it fits these two molecular weight species. If you'd like to fit everything globally, then it fits all three data sets at the same time to the single model, which is described over here. The molecular weight and the sedimentation coefficients, the molecular weight and the model parameters are shown over on the left-hand side. Parameters over here are relevant to this particular experiment. So suppose you did the sedimentation equilibrium with a different loading than you did the sedimentation velocity. They're different loaded concentrations. That's fine. Sedphat will deal with that as an individual parameter that's fit within only experiment 2 or only within experiment 3. But the global parameters are things like these molecular weights. And so then you can see, if I wanted to publish something kind of cool, I have three pieces of data that suggest I know what my molecular weight is in this mixture. There's 4 kilodaltons and 18.4 kilodaltons of stuff in this mixture. And I have DLS that probably gives me information on the big stuff. I have size exclusion chromatography or sedimentation equilibrium that gives me information about sort of everything, and sedimentation velocity that gives me information about the two separate species. So really the power here is that by multiplexing different types of experiments, you can see different types of, you have a justification in rendering more and more complicated models, if that makes sense to people. Normally I don't do modeling this complicated. Most of the time I just load in, say, 3 experiments or sometimes 6 experiments of sedimentation equilibrium data and run through a series of measurements that way, if that makes sense.
Um, for like the heterodimeric sort of like complexes or interactions, your concentrations that you're changing, that it's not the ratio of the two, it's the absolute amount of the two.
It's really tricky in those. If you pick one of these things, like a simple model, it's gonna crash on me. Super cool. You have to read the instructions closely and the help files will help a little bit. In some instances, depending on the model you pick with sedphat, it wants ratios and a total concentration. In other cases, it wants individual concentrations. And you have to be careful with it because in some places sedphat uses micromolar, in other places it uses molar. And it's usually obvious, but you should be aware that it's not always 100% consistent. They almost never use nanomolar for any of the concentrations, but it's something to be aware of. And in some cases, it does work better to fit things based off of a ratio. I mean, in terms of the mathematics of it, it makes more sense. So it's actually very powerful software to use that way that goes above and beyond using analytical ultracentrifugation. Although it crashes your computer an awful lot, as you may have seen. Are there other questions? I was going to kind of leave it at that as sort of like a bunch of introductory stuff. I didn't really find any interesting sedimentation equilibrium data that I have from way back when. I don't know if there's anything in 292. Yeah, it looks okay. But I never really saved it right with the right species. So I'm just gonna mess around a little bit and see if I can find some other pieces of data that look interesting. Very unlikely that I will because I kind of went through some of the more interesting stuff. Oh, Maya had anything here. Did we ever do anything with this? No, we didn't. Otherwise, please ask questions. Huh. I hope this was useful and interesting. I will, like I said, I'm around and I'm going to be answering people's emails. A couple people asked me about using the equipment. I need to get a little bit ready for the semester, but I should have some time off and on in between classes where I can help out getting some of these things spinning and getting some people some data. I will look at the data that I set up yesterday when you saw me put things up there and load material into the cells and get it ready to be spun. Like I said earlier in the morning, I came back and I spun things um, to see how it would look in the old machine when it was trying to do 3 wavelengths. It probably was a pretty miserable experience for the instrument, and we'll see how well we actually end up getting data for that. In the meantime, I will probably put out a survey and email everyone because I've been uploading these videos, and to respect people's privacy, I've put a password on it. The password is Optima AUC again, and so that would be the password for the new AUC workshop website. We'll be doing this again and hopefully some more advanced stuff in summer. I'll try to cut out some of the introductory material because I figure that the videos will allow people to catch up as they need. Otherwise, I was going to end things here and thank you for your participation. And please reach out if you have any questions or if you want to talk about setting up an experiment, or even better, if you have like data you've been struggling with and you want to throw your computer across the room because sedphat crashes on you twice a day. And as you saw, it crashed on me 3 times in about 35 minutes. Other questions? Well, I'm going to stop recording.